Biochemistry of muscle contraction, the sliding filament theory. Skeletal muscles in your body are attached to bones via tendons. These connections allow you to move your body when you contract a muscle. The first step in a muscle contraction starts in your brain, which sends a signal to your muscle. Each skeletal muscle is made up of bundles of muscle fibers called fascicles. Each of these fascicles is made up of individual muscle cells, which are called muscle fibers, or myocytes. The signal from your brain moves to the myofibers along motor neurons. When it gets to the muscle, it causes calcium to be released into the sarcoplasm, which travels in the brain through the transverse tubules. Each myocyte is then made up of individual myofibrils. These myofibrils are made up of repeating units called sarcomeres. Sarcomeres are the functional unit of muscles and are responsible for causing muscle contraction. A sarcomere is made up of thick and thin filaments called myosin and actin. These filaments are the moving parts of the muscle. The thin filament, actin, also has two other proteins attached to it called troponin and tropomyosin. When calcium is released into the muscle cell, it binds to troponin, which causes tropomyosin to change its shape. Because tropomyosin changed its shape, actin can now bind with myosin, which is the beginning of muscle contraction. In the first step, myosin heads split ATP and become reoriented and energized. The second step, myosin heads bind to actin, forming cross bridges. Then myosin heads rotate towards the center of the sarcomere, and finally, as myosin heads bind ATP, the cross bridges detach from actin. A muscle contraction begins when an action potential stimulates the release of calcium into the muscle cell. Calcium binds to troponin, which causes tropomyosin to change shape. This allows myosin to bind to actin. Once attached, the myosin heads can attach to actin and pull the ends of the sarcomere to the middle. When there is enough ATP and calcium present in the cell, the myosin heads can detach and reattach to actin to continue pulling actin in. This animation shows the sliding filament theory at work. The pink structures are the myosin filaments, while the light blue structure are the actin filaments. The next animation shows how important ATP is to the process of the sliding filament theory.